Hi everyone. Today we are going to talk about the fate of intermediates, energetics, and regulation of glycolysis. So we studied glycolysis in yesterday's class already. By lysis we mean breakdown, and by the word glyco it means glucose, breakdown of glucose. Revising breakdown of glucose, which is glycolysis, it can be aerobic or it can be anaerobic. The end product of aerobic glycolysis is pyruvate and anaerobic glycolysis is lactate. However, all the intermediates can be used up in various pathways. Let's have a look at that. So, coming to the first intermediate, when the glucose is phosphorylated by the hexokinase or glucokinase to form glucose 6-phosphate, this glucose 6-phosphate can be isomerized to a glucose 1-phosphate. Glucose 1-phosphate in turn forms UDP glucose and forms glycogen, which is the process of glycogenesis, formation of glycogen. Genesis means formation. Glycogen itself means glycogen, formation of glycogen. On the other hand, this glucose 6-phosphate can go into the pentose phosphate pathway, which is also called the hexose monophosphate shunt, in which ribose sugar is being formed. The ribose sugar can be used up for formation of nucleotides. The fructose 6-phosphate can form amino sugars and in turn lead to the formation of glycolipids and glycoproteins. Right? Coming to the split product, now we all know that fructose 6-phosphate had converted to fructose bisphosphate. However, fructose bisphosphate is a 6-carbon compound, but it will split into 2 3-carbon compounds, one aldehyde, glyceraldehyde 3-phosphate, and one ketone, dihydroxyacetone phosphate. What are these sugars? These are triose sugars. Glyceraldehyde 3-phosphate is a aldotriose, and dihydroxyacetone phosphate is nothing but a ketotriose. Now this ketotriose itself, of course, converts itself to, with the help of an isomerase, it's being catalyzed to glyceraldehyde 3-phosphate and continue into the glycolytic pathway. However, it can also form glycerol 3-phosphate, which is being used up in the formation of triacylglycerol, which is nothing but the storage form of lipid. The 1,3-BPG that is formed from the glyceraldehyde 3-phosphate, that is the 1,3-bisphosphoglycerate, can form 2,3-bisphosphoglycerate via the Rappaport-Lubering shunt. This 2,3-bisphosphoglycerate binds to deoxyhemoglobin and reduces the affinity of hemoglobin for oxygen so that maximum amount of oxygen is available to the peripheral tissues in conditions of high altitude. Why? Because in high altitude, there is less oxygen already in the air. Now, in such a state, do I want my oxygen to be binding more to the hemoglobin? Of course not. I want it to be released so that it can be utilized by the tissues, right? So if I decrease the affinity of hemoglobin, it's going to facilitate better delivery to the tissues. And who does that? That is, of course, done by this 2,3-BPG, which is formed in the Rappaport-Lubering shunt. The 3-phosphoglycerate can form serine. Phosphoenol pyruvate can form aromatic amino acids, and pyruvate itself can form aspartate with the help of the transamination reaction and the aminotransferase, aspartate transaminase, or it can form alanine with the help of the alanine transaminase. Transaminases are also called as aminotransferases because they transfer what groups? What functional groups? Basically, amino functional groups. And aspartate, of course, can be used up for pyrimidine formation and asparagine formation. Seen in this first slide that this fructose 6-phosphate forms fructose 2,6-bisphosphate with the help of the enzyme phosphofructokinase 2. Phosphofructokinase 2 forms fructose 2,6-bisphosphate. This 2,6-bisphosphate stimulates the phosphofructokinase 1. Okay, and this phosphofructokinase itself is being stimulated by insulin. As and when we have food, glucose is being absorbed into our blood, glucose level rises, and so is insulin released. This insulin will go and activate the phosphofructokinase 2, which in turn produces 2,6-bisphosphate. 2,6-bisphosphate goes and stimulates the PFK1, phosphofructokinase 1, and the glycolytic pathway sets on. Coming to the enzyme hexokinase in particular, hexokinase and glucokinase. Though we think of them as two separate enzymes, actually both are isoenzymes, you can say. Hexokinase is a blanket term, you can say, a kinase that works on hexose sugar. Okay, kinase is an enzyme that adds phosphate, and hexokinase meaning a kinase that adds phosphate to the hexose sugar. Now, this hexokinase usually has around 4 isoenzymes, 1, 2, 3, and 4. When I say hexokinase in general, I mean hexokinase 1, which is present in the peripheral tissues. To be specific, hexokinase 4 is also called as glucokinase. So glucokinase is nothing but hexokinase 4, which is exclusively present in the liver and beta cells of the pancreas. Now, if hexokinase 1 is present in the beta cells, hexokinase 1 is present in the peripheral tissue, yes, and hexokinase 4, which is also called as glucokinase, is present in the liver. Which of the hexokinase do you think will have a higher affinity for glucose? Let me tell you, it is hexokinase 1. Why? Because when I say peripheral tissue, it also includes red blood cells. Red blood cells are cells that utilizes only glucose as its fuel. So whether I am fasting or whether I am well fed, okay, in any state, red blood cells can use only glucose. If hexokinase 1 has low affinity for glucose, will it be able to use glucose when the concentration is very low? Of course not. So that is why I need a high concentration of glucose. That is why I need hexokinase to have a high affinity for glucose. Okay, by hexokinase I mean hexokinase 1. Now, why? Because when I'm fasting, I'll have low blood glucose. In low blood glucose also, I want my RBCs to have its energy, okay, which are nothing but considered peripheral cells or peripheral tissue cells. So, if it has high affinity, means it has lower Km value. Km value is what? In enzyme kinetics, Michaelis constant. It's the substrate concentration at which the enzyme is half saturated. Okay, and glucokinase, the hexokinase 4, which is present in the liver and beta cells of the pancreas, has much higher Km value and a low affinity. Why? Because when I have low glucose in the body, like say in fasting state, I don't want my liver to use up my glucose. I don't want my pancreas to take up glucose and get stimulated and release insulin, of course, when I'm in a fasting state, when I'm in low glucose state, isn't it? So what do I need? I need lower affinity of this enzyme so that the liver can use up the glucose from the blood only when glucose is very high, and my pancreas beta cells can also take up glucose and release insulin when the glucose levels are high, because I don't want my body, my liver, to use up glucose when it's less. So if I have to do that, I need an enzyme that can help in glycolysis in the liver as well as the pancreatic beta cells, that will have lower affinity for glucose, and that's why it will work only at a higher concentration. That's why it will have a higher Km value. So hexokinase, meaning hexokinase 1, is present in most tissues. Glucokinase means hexokinase 4 is present mainly in the liver and beta cells of the pancreas. This has low Km value, meaning higher affinity. Glucokinase has higher Km value, meaning lower affinity. It will work at higher concentrations of glucose. Vmax is low. Maximum velocity. Maximum velocity is higher. This can be inhibited by, hexokinase can be inhibited by glucose 6-phosphate, but glucokinase cannot be inhibited by glucose 6-phosphate. And one other feature is that hexokinase cannot be induced, but glucokinase can be induced by what? By insulin. Okay? Especially the amount that is present in the liver. What do you mean by inducer? A inducer, when I say inducer and repressor in particular, it has its effect on transcription. Okay? Insulin per se can affect the transcription of glucokinase and increase its amount, not just the activity. If I say plainly as an activator and inhibitor, if I have 5 molecules of enzyme, the activity might be increased. Maybe it becomes more faster, maybe it can act more faster, catalyzing capacity increases. But when I say inducer, say I have 5 molecules of glucokinase already, but my gene, at the gene level, the gene expression is getting induced and more glucokinase is being produced. Okay, instead of 5 maybe, now when it's induced by insulin, I'll have 10 molecules of glucokinase. So hexokinase is not something that can be induced by hormone insulin, whereas glucokinase can be induced by the insulin. Now, hexokinase, where is it mostly used? It's used in the peripheral tissue, like the red blood cell, mainly in the red blood cell, you can say now. So, if hexokinase is deficient, that means glycolysis is deficient in red blood cells, right? No energy for red blood cells, so red blood cells can get destroyed. That's why in hexokinase deficiency we'll have hemolytic anemia. But glucokinase is present mainly in the liver and beta cells of the pancreas. That means, what is it doing? It is regulating the blood glucose levels. Level of glucose present in the blood. Yes? So, if there is a glucokinase defect, that means the patient will present with what? With difficulty in regulating blood glucose. That means diabetes mellitus will happen. Okay. because hexokinase is involved in maintaining intracellular glucose concentrations in cells like RBCs when blood glucose is low, and glucokinase is involved in maintaining blood sugar. Okay, so deficiency of glucokinase or malfunction in glucokinase due to genetic mutations will lead to diabetes mellitus, and defect in hexokinase will lead to what? Destruction of the red blood cells, leading to hemolytic anemia. Okay. Now let's see the action of glucokinase or hexokinase 4 a bit in detail. When glucose is elevated in the blood, right after our food, you say, glucose is being absorbed, so it's more in the blood. So it is going to be taken up by the GLUT2 transporters, okay, which is sensitive to the concentration of glucose. It's going to take it up. As soon as it takes it up, this glucose is going to stimulate the release of hexokinase 4 from its regulatory protein. Hexokinase 4 or glucokinase normally is bound to a regulatory protein subunit. When it is bonded to this regulatory protein, it cannot function. But once it gets detached from the regulatory protein, it becomes active. So this glucose will stimulate the release of hexokinase 4, and it will form glucose 6-phosphate and fructose 6-phosphate. This inactive complex stays in the nucleus, you can see, near the nuclear membrane. And once stimulated, only the hexokinase catalytic part comes into the cytosol. Okay? And produces glucose 6-phosphate, which is isomerized to fructose 6-phosphate. This fructose 6-phosphate can stimulate the binding of hexokinase to the regulatory protein and decrease the activity of the pathway whenever needed, because the pathway flows from glucose to glucose 6-phosphate to fructose 6-phosphate. Glucose stimulates the active formation, okay, and stimulates this pathway. But towards the productant, 6-phosphate is formed. 6-phosphate cannot inhibit this, as we saw earlier. But fructose 6-phosphate can enhance the binding of this hexokinase to its regulatory protein. Okay, and bring down its phosphofructokinase, as we saw, a high energy state. It shows here what? A high energy state is going to inhibit it. Okay, and a low energy state is going to stimulate it. And also, I told you, fructose 2,6-phosphate is going to stimulate it. Okay, a low energy state and fructose 2,6. A high energy state is going to inhibit it, and also the citrate of the TCA, and a low pH is also going to inhibit it. Okay, this diagram signifies the same thing again. So this you can read through, which I have already explained. Now, this fructose 2,6-bisphosphate stimulates PFK1, right, to form fructose 1,6-bisphosphate. But at the same time, this molecule, this compound, inhibits the activity of fructose bisphosphatase, the enzyme which is capable of converting this 1,6-bisphosphate to the 6-phosphate form. Okay, it can remove, this enzyme can remove a phosphate from here and convert it to a single fructose monophosphate form. But if I want my glycolysis to proceed, I don't want the reverse reaction happening. So this phosphatase enzyme also has to be inhibited. Okay, and this inhibition is done by fructose 2,6-bisphosphate. One side, it stimulates phosphofructokinase so that 6-phosphate, fructose 6-phosphate can become 1,6-bisphosphate, and on the other side, what it does, it prevents the conversion of this back. And that's how it regulates. Okay. Actually, this fructose bisphosphatase activity is present in the PFK2 molecule itself. PFK2 molecule, the phosphofructokinase 2 molecule, is a bifunctional enzyme. It can form fructose 2, a part of it can form fructose 2,6 bisphosphate, and a part of that enzyme can act as the bisphosphatase. Okay, so it inhibits the fructose 2,6 bisphosphate is produced by the PFK2, and it inhibits the phosphatase activity of that same protein. I hope you know that some proteins are multifunctional. They are called multifunctional enzymes or multifunctional proteins. So phosphofructokinase 2 is one such enzyme which has a 2,6 bisphosphate activity as well as a bisphosphatase activity. So coming to pyruvate kinase is an enzyme which is active on dephosphorylation. I hope you all know that some of the enzymes are capable of being active on being phosphorylated and some are deactivated on phosphorylation. Similarly, some enzymes are active on dephosphorylation and some enzymes are inactive on dephosphorylation. So pyruvate kinase is an enzyme which is active on dephosphorylation. Okay, and when it is phosphorylated, it is inactive. But who phosphorylates this pyruvate to make it inactive, or who dephosphorylates it? Let us see. Now, remember, insulin is a hormone which usually tends to dephosphorylate. Just remember this general concept. Insulin tends to dephosphorylate and glucagon tends to phosphorylate. Now, right after you have food, which hormone would be more, insulin or glucagon? Right after I have food or you have food, blood glucose levels would be high. So insulin would be high. So when insulin is high, insulin would lead to dephosphorylation of the pyruvate kinase. How it dephosphorylates? It dephosphorylates by acting through the tyrosine kinase receptors that leads to activation of protein phosphatases. A protein phosphatase is capable of phosphorylating enzymes such as pyruvate kinase. So it's going to remove the phosphorus group or phosphate group from the pyruvate kinase and render it active. Making it active, meaning phosphoenol pyruvate would form pyruvate. Yes, and in this process, of course, a ATP is being formed, isn't it, in the pathway? It's an energy producing step. Now, when our blood glucose levels are low, when we are fasting, say overnight fasting, blood glucose levels are low, will my insulin be high? Of course not. My glucagon would be released so that the blood glucose level can be maintained. Yes, glucagon or the anti-insulin or hyperglycemic hormones would be more in the blood in order to maintain the blood glucose level in fasting state. Yes, that's why we have said low blood glucose level, meaning does not mean pathologically low, okay? Meaning in fasting. Say, in low blood glucose level, hyperglycemic hormones such as glucagon would be high. Glucagon would phosphorylate this pyruvate kinase. How does it phosphorylate? By acting through the cyclic adenosine monophosphate pathway, the CAMP pathway, which CAMP is nothing but a second messenger. How is this CAMP formed? Glucagon binds to a G protein coupled receptor. Okay, it's a G protein coupled receptor, which is a 7 subunit transmembrane protein. It will bind to this receptor and it will activate a adenylyl cyclase. Adenylyl cyclase will convert a ATP to the cyclic AMP. This cyclic AMP in turn goes and activates protein kinases. Okay, this protein kinases can in turn go and phosphorylate enzymes like pyruvate kinase. Protein kinase meaning adding phosphate to a protein. Which protein? Pyruvate kinase. Enzyme is nothing but a protein. So if this is phosphorylated, this will become inactive. Okay. So high blood glucose, high insulin, dephosphorylation, pyruvate kinase is active, glycolysis proceeds. Low blood glucose level, fasting, high glucagon, high hyperglycemic hormones or anti-insulin hormones, phosphorylation happens by the CAMP pathway and pyruvate kinase is less active and glycolysis is inhibited. Okay. Other than that, arsenate and iodoacetate are inhibitors of the glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate dehydrogenase enzyme. This enzyme has a cysteine residue in its active site, which forms thiohemiacetal with the glyceraldehyde group, aldehyde group of the glyceraldehyde 3-phosphate. Okay, and that formation of thiohemiacetal is prevented because this heavy metal like arsenic or arsenate and iodoacetate compounds during poisoning, they bind to the cysteine. Cysteine has what? Sulfur. And anything with sulfur can be represented by what? Thio. Coming to fluoride. Fluoride inhibits the enolase. How? It inhibits enolase by formation of a fluorophosphate, which forms a complex with the magnesium ion, okay, that is being used by this enzyme at the active site of this enzyme. Now, do you know about PET scan, which is being done to detect cancers? What happens in cancers? There is more cell growth than normal. Hyperplasia and hypertrophy, more number of cells, bigger cells, right? So if a tissue has more cells than normal, it will also need more fuel, more food, right? So normally, if a cell has 1 unit of glucose, this cancerous cells will have 10 times more, you can say. Okay, it's common sense. Cancer cells, tumor, big growth, right? More number of cells, bigger in size, more food is needed, more fuel is needed. Glycolysis will be more. Okay, glucose uptake would be more. So when glucose is given to these cancer cells, it uses more. Based on this, PET scan was developed. In PET scan, they use fluorodeoxyglucose. Okay, that is, the glucose molecule is deoxygenated and it's fluorinated. Okay, when this deoxyfluoroglucose or fluorodeoxyglucose is being taken up by the tumor cells, the hexokinase can work on it and phosphorylate it, just like glucose is being phosphorylated. Okay, it can phosphorylate it at the 6th carbon. But after phosphorylation, because the fluoride is here, the next step of the reaction, isomerization to fructose, cannot happen. Okay, so the phosphohexose isomerase cannot work. Now what happens? This molecule accumulates inside the cells, right? Inside the tumor cell. That gives the signal which is being captured as signals in PET scan. And more the signals we get, worse the tumor, of course. And that indicates tumor aggressiveness and a poor prognosis. Now, hexokinase deficiency, we saw hemolytic anemia. How does hemolytic anemia happen in hexokinase deficiency? Now say hexokinase is deficient, glycolysis won't work. Glycolysis won't work meaning there is no energy. No energy, what will happen? The sodium-potassium pump, which is dependent on ATP, will get disrupted. And when sodium-potassium pump gets disrupted, the spherical structure of the membrane of RBCs will get disrupted. Why? Because the membrane potential would get disrupted because of the malfunctioning of the sodium potassium pump, which is dependent on ATP, because ATP will not be there because of lack of glycolysis. In turn, there is deficiency of energy and disruption of membrane potential and breakdown of the cell membrane of RBCs. Okay, and when the cells break down, who has to scavenge it? The reticuloendothelial system, meaning the spleen. And the hemoglobin that was inside the cell, that would also be degraded. When heme of hemoglobin is degraded, that will lead to increased bilirubin level, hyperbilirubinemia. Okay, and of course, red blood cells will have decreased hexokinase activity, and when more cells are being broken down in hemolytic anemia, that means the body has less RBCs. Bone marrow will produce more red blood cells, and in the process it will release immature cells into the blood, and that's why there would be increased reticulocytes, and of course destruction of RBCs is there, and less hemoglobin. Less hemoglobin meaning the adult hemoglobin will be less. Okay, A2 hemoglobin will be less. Body will try to compensate, bone marrow will try to compensate, forming fetal hemoglobin. Okay, so that would be mainly anemia, jaundice, because heme would be degraded to form bilirubin, more heme, more RBCs destroyed, more hemoglobin destroyed, more heme would be degraded, more heme degradation, more bilirubin formation. More bilirubin formation is nothing but jaundice. And where does the rest of the remnants go? It goes to the spleen, right? To be scavenged by the reticuloendothelial system. Spleen works more. There would be splenomegaly. This is a autosomal recessive condition. Okay, that means two copies, two alleles has to be defective. It has to be a biallelic homozygous mutation in the hexokinase 1 gene or a biallelic compound heterozygous mutation. Coming to the hexokinase 4 glucokinase gene deficiency, we already know that it's present in the liver and beta cells of the pancreas. Now, this is also an autosomal recessive disorder, meaning biallelic homozygous or compound heterozygous mutations will lead to this disorder. Now, if glucokinase is deficient, of course, blood glucose cannot be maintained well. Yes, the glucose from blood cannot be utilized. That means there would be high blood glucose, hyperglycemia. If there is high blood glucose, lot of sugar, lot of glucose in the blood, the proteins will tend to get glycated. Okay, now hemoglobin, globin is a protein. Okay, this hemoglobin will get glycated, and that is nothing but hemoglobin A1C, glycated hemoglobin. At the same time, what will happen? The beta cells of the pancreas will not use this glucose. If glucose stimulation is not there from the side of the glucose, insulin will not be released. Insulin is not released, insulin is not produced means what? Of course, the proinsulin is not produced. Maturation of proinsulin to insulin gives C-peptide in the blood. Okay. Now, if maturation of proinsulin to insulin is not there, that means C-peptide levels in the blood also will be less. C-peptide is less when production of insulin is less. Okay, even on stimulation with glucagon, C-peptide levels will still be less. But however, the plasma glucagon would be normal and antibodies against insulin or glutamic acid decarboxylase would be negative. Why we are looking for antibodies here? To differentiate it from type 1 diabetes, regular type 1 diabetes, which is autoantibody mediated. When we don't see antibodies and we see low serum C-peptide levels, normal plasma glucagon, and there is diabetes in the neonate, we would think of what? A glucokinase gene defect. Okay, and because Glucose cannot be utilized by the body. In utero, the child will have intrauterine growth retardation. And of course, if there is intrauterine growth retardation in utero itself, the baby will be born with low birth weight and will be smaller in size, small for gestational age, and present with nothing but neonatal diabetes mellitus. Coming to pyruvate kinase deficiency. Again, pyruvate kinase deficiency also, it leads to hemolytic anemia. This is also autosomal recessive disease. Okay, and the gene is the red cell pyruvate kinase gene. In this also, because pyruvate kinase will not work, there would be no energy, no glycolysis, low levels of ATP, leading to dysfunction of the sodium-potassium ATP pump and membrane potential being disrupted, and there would be hemolytic anemia, lysis of the red blood cells. Lysis of the red blood cells would be there. Okay, and because of the lysis of the red blood cells, what will be there? High bilirubin, because heme of hemoglobin will be also broken down, and high bilirubin will lead to jaundice. Okay, and in the gallbladder, because of the high bilirubin, there would be cholelithiasis and cholecystitis. Yes, and splenomegaly, because spleen is scavenging. Okay, in prenatal manifestation, rarely there would be intrauterine growth, and there could be edema of the whole fetus, which is called as hydrops fetalis. Hydrops meaning water, fetalis meaning of the fetus, and it's non-immune mediated. But these are non-specific findings again. So thank you. We end our class here today.